welcome to the genetic reassortment narration. So we're going to begin by making a model of human or seasonal influenza virus. So we're going to use the cup, and this cup represents the capsid of the human flu virus. We're going to put 6 of the fuzzy stems, 1 blue sparkly stem, and 1 blue twist tie into the cup. So let's begin. All right. These represent the RNA strands, or genetic material, that determine what proteins the human flu virus can make. Next, we're going to attach 3 blue circle stickers to the outside of the cup. And I'm going to go ahead and put them on the back of the cup since we already have the label in the front. All right, and these blue circle stickers represent the antigens, or proteins, on the surface of the human influenza virus. So you can set this to the side. Next, we're going to be making the avian or bird influenza virus. So we're going to use the cup labeled as such, and this cup represents the capsid of the avian flu virus. We're going to put 6 red fuzzy stems, 1 sparkly red stem, and 1 red twist tie into the cup. These represent the RNA strands, or genetic material, that determine what proteins the avian flu virus can make. Next, we're going to attach 3 red heart stickers to the outside of the cup. These red heart stickers represent the antigens or proteins on the surface of the avian flu virus. So same thing, we're just going to set this to the side. And then next we're going to make a model of the swine or pig flu influenza virus. So we're going to use the cup labeled as such, which represents, again, the capsid of the swine flu virus. And we're going to put 6 green fuzzy stems, 1 green sparkly stem, and 1 green twist tie into the cup. These represent the RNA strands, or genetic material, that determine what proteins the swine flu virus can make. Next, we're going to attach 3 green star stickers to the outside of the cup. These green star stickers represent the antigens or proteins on the surface of the swine flu virus. All right, so next we're going to set up the host cell. The host cell, which is a cell in the lining of the pig respiratory system, we're going to be using the sheet that you were given in your kit called Host Cell, Cell Lining in Pig Respiratory System. Here it is. We're going to put the pink foam tray, or ours is white in this instance, in the center of the host cell sheet. The pink foam tray represents the nucleus of the pig cell. Next, we're going to begin our attachments. So when an influenza virus infects a host cell, it attaches to the receptors on the surface of the host cell. We're going to put the avian, human, and swine virus models on the half circles located at the top of the host cell. Match the antigen stickers on the virus models with the shapes in the host cell surface. So as you can see, the stars match these. The circles match these, and the hearts match these. So there we go, all nice and lined up. The next step is penetration, which is when the attached virus enters the host cell. we're going to move the 3 virus models, human, pig, and bird, into the host cell. The next step is uncoding. The RNA or genetic material from the virus is released and enters the nucleus when it takes control of the host cell. So we're going to empty the RNA from the human, bird, and pig models into the host cell, which is, once again, the white tray. So, just going to dump them out. Great. So the next step is biosynthesis. The virus RNA then detects the host cell to make new viruses. So we're going to replicate or copy the RNA. So we're going to add one of each of the fuzzy stems, sparkly stems, and twist ties to the pink foam tray. All right, so I'm going to start with the green again. So we're going to add the remaining 6. 1, 2, 3, 4, 5, 6. One of these. And one of these. Next, we're going to do the red. One of these. And one of these. And then finally, we're going to do it again for the blue, which is the human. 3, 4, 5, 6. 1. And 1. So the next step is assembly. The viral parts, meaning the RNA, capsid, and surface proteins, assemble to make new viruses. So we're going to make 3 new viruses. We're going to start by placing the 3 new influenza cups, which are these, at the bottom of the host cell. So that's over here, and the number on here corresponds to where they're going to be meant to go. So 3, 2, 1. All right, for each cup, we're going to randomly, meaning without paying attention to the colors, pick up these items from the host cell nucleus pink tray and place them into each of the cups. So we're going to grab 1 twist tie, well, that's 1 sparkly stem, but now we're going to grab 1 twist tie. And then we're each going to grab 6 fuzzy stems for each of the cups. So 1, 2, 3, 3, 4, 5, 6. And I may move this tray out of place because I am trying not to look. 3, 4, 5, 6. And 1, 2, 3, 4, 5, 6. All right. We're going to use the colors of the fuzzy stems to determine which sticker colors should be attached to the outside of each new influenza cup. Then attach 1 color sticker for each different color on the fuzzy stem. Notice, there are only 3 colors of stickers, so you should not need more than 3 stickers on each cup. So for this cup, we have 1 of each color. So we're going to put a sticker on for 1 of each of the colors. So let's start with green. We have some green. We definitely have blue. And then we also have red. And then the same thing applies for this one. So we also have all colors. For all of these, I actually managed to grab 1 of each color. So we're going to put 1 of each color onto each of these cups. And again, like they said, you should have no more than 3 stickers in the cup because there are only 3 colors to choose from. The next step is release. So the newly assembled virus particles are then released from the cell. We're going to place the 3 new viruses in the circles located at the bottom of the pig cell, which I had messed up and I had already put them there, but essentially you're going to take all 3 of these and put them to their corresponding thing. So once again, this is number 3, this is number 2, and then this is number 1. And there you have it. That's your genetic reassortment model. Thank you for listening.